Welcome to your first semester of cell biology here at Delhi University. Today, we are going to tackle a fascinating puzzle. Every single cell in your body contains about 2 meters of DNA. How does the cell fit all of that information into a microscopic nucleus without it becoming a tangled, unreadable mess? The answer lies in the dynamic packaging system of euchromatin and heterochromatin. Let us dive in and see how this works. To understand this packaging, imagine you have a massive, sprawling library containing millions of instruction manuals for building a city. If you left every single book open and scattered across the floor, the library would be an unnavigable, chaotic mess. Instead, clever librarians pack away the books that are rarely used into tight, dense storage boxes. Meanwhile, they keep the books that are needed every day open on the reading tables. In your cells, your DNA is exactly like this massive library. The cell must pack it up to save space, but it also needs to read certain genes constantly to keep you alive. Now that we have our library analogy, let us look at the actual biology. In the nucleus of eukaryotic cells, DNA does not exist as a naked, floating double helix. Instead, it is wrapped around specialized spool-like proteins called histones. Together, this complex of DNA and proteins is known as chromatin. Think of histones as the physical bookshelves and boxes in our library. Depending on how tightly the DNA is wound around these histones, chromatin exists in 2 primary forms. These 2 forms dictate whether a gene is actively being read or if it is packed away in storage. This brings us to the first form of chromatin, known as euchromatin. Remember the open books on the reading tables from our analogy? That is euchromatin. It is the lightly packed, less condensed form of chromatin. Because it is loosely coiled, it is rich in active genes. The cell is actively reading its genetic code to make proteins, a process called transcription. Because the DNA is so open, bulky cellular machinery, like the enzyme RNA polymerase, can easily physically access the DNA sequence. This is the active working state of your genetic material. In contrast to those open books, we have heterochromatin, which represents the tightly packed storage boxes. Heterochromatin is the highly condensed form of chromatin. It typically contains inactive genes or structural DNA regions, such as telomeres and centromeres, which do not code for proteins. Because this chromatin is so densely compacted, the transcription machinery simply cannot physically reach the DNA. As a result, these regions are rendered transcriptionally silent. The cell has effectively locked this information away because it doesn't need to read it right now, saving valuable space in the nucleus. Understanding these 2 states is absolutely crucial because it explains a fundamental mystery of biology. How can a neuron in your brain and a muscle cell in your leg do completely different jobs when they contain the exact same DNA? The answer is differential packaging. The muscle cell packs away the neuron-specific genes into dense heterochromatin, while keeping its own muscle-specific genes open as euchromatin. If our cells could not form heterochromatin, every gene would be active all the time, leading to cellular chaos and disease. Furthermore, tightly packing DNA acts as a physical shield, protecting our most vital biological information from dangerous mutations. Before we move on to the mathematics of packaging, let us visualize this inside an actual cell. Here, you are looking at a cross-section of a nucleus. Notice the outer boundary, the nuclear envelope, dotted with pores. Inside, you can clearly see the two states we just discussed. In the center, the lighter, wavy threads represent euchromatin. The top inset shows how loose this is, allowing RNA polymerase to attach. Now look at the dark, dense clumps pushed against the inner edge of the nucleus. That is the heterochromatin. The bottom inset reveals how tightly the nucleosomes are clustered together, making it impossible for enzymes to enter. This spatial organization is key to gene regulation. Now that we have seen the physical structure, we can quantify exactly how tightly the DNA is packed using a mathematical concept called the compaction ratio. The formula is C equals L DNA divided by L chromatin. Let us break down these symbols. C is the compaction ratio itself, which is a dimensionless number telling us how many times the DNA has been folded. LDNA represents the total physical length of the naked DNA double helix if you stretched it out in a perfectly straight line. L-chromatin represents the actual measured length of the folded chromatin structure. When the chromatin is packed tightly into heterochromatin, L-chromatin decreases, which causes the compaction ratio C to increase dramatically. Let us apply this formula with a concrete example. A scientist is studying a specific gene. When fully stretched out, the naked DNA has a length of 3,400 nanometers. When the cell is actively reading this gene, it exists as loosely packed euchromatin with a measured length of 100 nanometers. We want to calculate the compaction ratio. We start with our formula, C equals L DNA over L chromatin. We substitute 3,400 for the stretched length and 100 for the folded length. Dividing 3,400 by 100 gives us 34. This means the DNA is compacted 34 times. This relatively low number makes perfect biological sense, as the DNA must remain loose enough for transcription machinery to attach. Now, let us see what happens when the cell stops needing this gene and packs it away. The stretched DNA length remains exactly the same at 3,400 nanometers. However, the new heterochromatin length is drastically reduced to just 10 nanometers. We use the exact same formula, but this time we divide 3,400 by 10. The result is 340. The DNA is now compacted 340 times, which is exactly 10 times denser than the euchromatin state. Physically, this massive increase in the compaction ratio explains why the transcription machinery can no longer access the gene. The DNA is simply too tightly wound, effectively silencing the gene. Now that we have used the formula to find the compaction ratio, let us look at how we can manipulate it to find unknown lengths. Often, scientists can only see the compacted form under a microscope and know the standard ratio, but they want to know the original stretched length. We start with our base formula, C equals L DNA divided by L chromatin. To isolate L-DNA, we multiply both sides of the equation by L-chromatin. The terms on the right cancel out. We then flip the equation to get our final result. L-DNA equals C times L-chromatin. This straightforward algebraic manipulation is incredibly powerful, allowing us to mathematically unfold the DNA and calculate dimensions that are impossible to measure directly. Let us put our newly derived formula to the test. During cell division, a region of heterochromatin at the centromere has a known compaction ratio of 1,000. Under a microscope, its length is measured to be 2.5 micrometers. What is the total length of the naked DNA? We take our rearranged formula, LDNA equals C times L chromatin. We substitute 1,000 for C and 2.5 for the chromatin length. Multiplying these together gives us 25,000 micrometers, which is 25 millimeters. This is a staggering result. A piece of DNA long enough to see with the naked eye is compressed into a microscopic package just 2.5 micrometers long. Without this extreme compaction, our cells would physically burst. We have covered a lot of ground today, so let us review the key takeaways. First, euchromatin is the open, accessible form of DNA that allows the cell to read genetic instructions and build proteins. Second, heterochromatin is the dense, tightly packed form that protects the genome and keeps unneeded genes turned off. Third, by constantly shifting specific regions of DNA between these two states, a single fertilized egg can develop into a complex human being with specialized cells. Finally, we learned how to mathematically measure this incredible biological packaging using the compaction ratio. You now have a solid foundation in how your cells manage their genetic library.